Eu começo, né?
Bom dia a todos. Por favor, Marcelo, você pode começar?
Bom dia. Eu acho que não sou eu que estou na tela.
É a tela
Bom dia a todos. Bom dia a todos. Eu sou o Marcelo Brígido, eu estou aqui substituindo a professora Andréia nessa apresentação, né? Hoje a gente continua nossos seminários e hoje a gente vai receber o professor Teesalu, da Universidade de Tartu, na Estônia, desculpe. O professor Ricardo vai fazer a apresentação do professor Tambet e após isso, o professor Tambet vai fazer a sua apresentação. Certo? Só estou lembrando aqui, a gente ficou uma semana sem seminário, a gente está tendo problemas para conseguir seminários de estrangeiros nessa época de verão, está todo mundo viajando. Então, a gente conseguiu o professor Tambet, que ele está trabalhando já voltou, né, para as férias de outono, né, para o período de outono. Então, assim, e eu só lembrando aí para vocês, né, dar like no YouTube, né, se inscrever no canal, e a gente vai montando essa comunidade aí de seminário que está muito interessante, né? A gente está sempre tendo novidades, seminários muito interessantes. Então, vou passar a palavra aqui para o professor Ricardo, que vai fazer a apresentação do professor Tambet.
Tambet, I'm going to introduce you in Portuguese first and then in English, all right?
Thank you.
Pessoal, bom dia a todos. É um prazer muito grande de apresentar a vocês o professor Tambet Teesalu. O Dr. Teesalu é o professor titular de nanomedicina no laboratório e chefe do laboratório de biologia do câncer da Universidade de Tartu, na Estônia. Além disso, ele também é professor adjunto da Sanford Burnham Prebys Medical Discovery Institute em La Loja, na Califórnia, nos Estados Unidos. Ele fez o doutorado dele na Universidade de Helsinki, na Finlândia, em 1999, e o principal foco do laboratório do Dr. Teesalu é o desenvolvimento de testes diagnósticos e terapêuticos, o que a gente hoje em dia chama de nanoteragnóstico, para detecção e tratamento de tumores sólidos, como glioblastoma, câncer de mama, entre outros. So, it's my pleasure to introduce you, Dr. Tambet Teesalu. Dr. Teesalu is full professor of nanomedicine and head of the cancer biology laboratory at the Tartu University in Estonia, and adjunct professor at Sanford Burnham Prebys Medical Discovery Institute in La Loja in California, United States. Dr. Teesalu received his Ph.D. from the University of Helsinki in Finland in 1999, and his main focus at his lab is the development of novel diagnostic tests and therapeutics for the early detection and precision treatment of solid tumors, such as glioblastoma, breast cancer, and others. So, Tambet, thank you very much for your kindness in presenting us your work. So, please.
So, good morning, everybody. Hope you can hear me well, and I'm really pleased to be talking on this meeting. And as Ricardo mentioned, I'm now based in Estonia, which is in north part of Europe. We still have summer here. The situation is not too bad, so life almost feels normal. And today I will give you kind of a big overview, not too detailed, but just to introduce you to the topic of precision targeting of cancer with tumor homing peptides. So this is basically some background on my training for you to understand also the way how I approach things. I was trained first in yeast molecular biology, then moved to mouse embryonic development, did my Ph.D. on mouse trophoblast implantation as a model of invasive process, and then moved more into cancer biology, did postdoc with famous cell biologist and cancer biologist Erkki Roslahti in the US, and then returned back to Estonia to start the lab of cancer biology. And I still do a lot of collaboration with the institutes I used to work in the States, Sanford Burnham and UCSB, with Erkki and Jamie Mart. And this is the outline that I'm going to discuss today. So first, I'll tell you about the concept of vascular zip codes and tumor-homing peptides. Then introduce you a novel class of tumor-homing peptides that we have discovered that we call tumor-penetrating peptides, with focus on peptide called IgD, or internalizing IgD peptide, that is now entering phase 2 clinical trial for pancreatic cancer targeting. And then bring you some examples of novel homing peptides, one targeting matrix in solid tumors, and then the other one targeting the cell surface molecule named P32 that is also upregulated in tumors. So the basic problem that we are trying to address is that there is a plethora of cytotoxic agents that are used in fight against cancer, but they lack, in most cases, intrinsic anti-tumor selectivity. They are doing a very good job in killing the cells, but unfortunately, this includes being damaging to healthy cells. So chemotherapeutics, they have this relatively narrow difference between toxic and efficacious dose, which is called therapeutic window. And one way to broaden this window is by using affinity targeting ligands such as peptides or aptamers. So with aim being more drug being delivered to tumor and difference between toxic and efficacious dose become broader. And of course, this would allow also to improve this dose distribution to have more drug reaching the tumor for more efficacy. And then the basis of such affinity targeting is a recognition that there is a system of vascular zip codes in our vasculature. Somehow, you know, in our studies at university, we are trained to think that endothelial cells are endothelial cells and they are relatively similar in tissues, but actually there are very important and clear tissue-specific differences in endothelial cells, including the part of endothelial cells that is accessible to systemic circulation. And this diversity, which is dependent on the tissue type and status, can be targeted with circulating probes, antibodies, peptides, aptamers, to enable delivery of specific imaging probes, development of precision-guided drugs, and actually to reveal druggable targets. This concept of vascular zip codes was first coined by my postdoc supervisor, Erkki Roslahti here. And he also developed a clever technique called in vivo phage display to explore this vascular heterogeneity. And I'm not sure how much you know about bacteriophage peptide display, but just I give you very minimal background so you understand what's going on. So bacteriophage is a bacterial virus, and its genome can be modified in such a way to display on its coat random peptides or even antibodies. And this is called peptide phage display. We like to use T7 phage, which has a size of typical clinically used nanoparticle. Its capsid diameter is 50 nanometers, and it's unlike the filamentous phage as aspect ratio that is also similar to clinical nanoparticle. And then to map the vascular diversity, you now take the peptide phage library. So a complex mixture of phages with about 1 billion of different peptides displayed on the surface of bacteriophage, and then inject this complex mixture into a living mouse bearing your target disease, in our case tumor. And then after circulation, then the pool of bacteriophages that display peptides with affinity to this tumor or other disease you are interested in preferentially accumulate in this disease, in this site. And then you just, after circulation, excise your tumor, extract the phages, and repeat the cycle. This is called biopanning. And then after a few cycles, you will start seeing predominant accumulation of peptides that stick to molecules in your target vessels. And then, of course, nowadays we have come a long way from this very basic approach. We use routinely next generation sequencing to look comprehensively at the selected phage pool and the shifts in peptide landscape and different bioinformatics tools to allow that. Now, this is an example that I always show and actually have people who join my lab to use with their own hands to convince them that indeed homing peptides work. And the experiment here is that you take 3 phages. One is a brain homing phage, the other is a lung homing phage, and then a negative control. You amplify them individually, mix together into equimolar mix, and then inject them into a mouse systemically, excise brain, lung, and liver, and then go and evaluate the representation of each phage after circulation. And as you see, as expected, the lung homing phage, which is R-bar, accumulates preferentially in lung, and brain homing goes to brain. Now, peptides as targeting ligands, as always, have certain advantages and disadvantages. Advantage really is that they are very biocompatible, of course, typically non-immunogenic. The size of peptides that we usually display on the phage is 9 amino acids. And interestingly, short peptides also typically bind to important sites on target molecules. And this means that those peptides that we select are often biologically active. And then finally, of course, peptides are cheap to manufacture and easy to scale up. Disadvantage of peptides is that they typically don't have very high affinities. Typical affinity of homing peptide to its receptor is about 1 micromolar. And then, of course, being peptides, they are prone to proteolytic degradation. But both these low affinity and instability issues can be dealt with tools that we have available nowadays. You can use amino acid modifications to improve the stability and proteolytic resistance, and also the low affinity is something that you can deal with, especially in nanoparticle delivery, by using multiple peptides for delivery and achieving high avidity of interaction. Then the binding site barrier is one important concept that is also has to do with affinity of targeting ligands. People have noticed for a long time that high affinity targeting ligands such as antibodies, they tend to accumulate at first met sites in vascular beds, whereas the lower affinity ligands such as peptides, they move deeper into tissue parenchyma, and that's called binding site barrier. So it means that not always high affinity means better drug delivery. And due to these advantages and ease of identification of homing peptides, or relative ease, I should say, it's not surprising that in many cases, papers that deal with nanoparticle targeting have chosen peptides as targeting moieties. Here is simply the number or representation of citations for this period, which shows that in more than half of the cases, the research reports rely on peptides for targeted delivery. Now, this was simply a basic introduction, and I would like to go more into specific peptides and bring you some examples. One peptide that I want to discuss is IRGD. It's a prototypic tumor-penetrating CN-rule peptide. And we discovered it together with Kazuki Sugahara, who is shown here also at the bottom, who was then the postdoc in Ökiro's Lahti lab and is now a physician scientist at Columbia University in the US. And really, the IRGD illustrates a concept of in vivo phage display and how useful it is for agnostic discovery of multi-stage tumor targeting peptide. So IRGD really uses a multi-step tumor homing and penetration mechanism depicted here in this schematic that ended up as a cover of Science magazine. IRGD is a cyclic 9 amino acid peptide that is depicted in this figure as a circle. And first it homes to angiogenic integrins upregulated in the tumor blood vessels. Then after this recruitment in pericellular endothelial cell surface, which is a proteolytically privileged site, there happens a proteolytic cleavage of IRGD to cut this peptide into this motif CRGDK that we call CN rule motif. This cleavage makes IRGD to fall off from angiogenic integrins and then bind to secondary receptor of the peptide, which is called neuropilin-1. And then this neuropilin binding engagement triggers the transcytosis pathway through endothelial cells and through tumor tissue to reach deep into tumor parenchyma. So basically we have here 3 steps: integrin homing, proteolytic processing, and neuropilin engagement, which would have been very hard to rationally design into the system. But the phage display allowed this to happen, and then we kind of reverse work to define the exact mechanism. And this is how well IRGD works. Here is a transgenic mouse model of pancreatic ductal adenocarcinoma. And if we inject those mice with phage that is displaying simply RGD peptide that simply sticks to integrins, but lacks this proteolytic processing and sender mechanism, then what we see is accumulation of the phage in the angiogenic vessels, and phage here is green and vessels are red. But no, it doesn't really make it outside the vessels or into tumor parenchyma. Whereas the phage coated with IRGD peptide first, of course, homes to vessels, but then over time it escapes blood vessels and penetrates into tumor parenchyma. So you can appreciate the difference in the extent of green signal here versus here. How can you take advantage of this peptide? Why can it be useful? First and most obvious example, of course, is you can use it for conjugated payload delivery. So basically you take the peptide, you chemically couple it to nanoparticles or drugs that you want to deliver and get improved tumor tropism and accumulation. And here is an example. Now, if we take iron oxide nanoworms, basically iron oxide nanoparticles that are MRI active, and then image them over time in mice. So here is pre-injection, and then after 3 hours with IRGD nanoworms, we have this increase in dark signal, and this persists over time. Whereas there is only a small increase in signal with CRGD-C, only integrin targeting nanoworms, and no change for untargeted nanoworms. And on your right, you can see also the validation of accumulation by confocal imaging, since the nanoworms carried also FAN label. And then a postdoc in my lab, Lorena Simon Garcia, tested a nano platform, which is pH-dependent polymersomes that we obtained from Giuseppe Battaglia's lab in UCL. And these particles appear to work very well when coated with IRGD in intraperitoneal delivery. As you may know, peritoneal carcinomatosis is a deadly condition where different abdominal tumors spread loco-regionally in the peritoneal cavity, and it's very hard to do anything for this condition. You can remove the tumor nodules, but it keeps coming back. So what is badly needed is a therapy that would allow development of a drug that would smartly recognize these nodules and eliminate them. And Lorena saw that IRGD polymersomes accumulated very nicely in the tumor nodules. Here is shown a gastric cancer, but we have tested the same also for colon and ovarian cancer. So we get nice accumulation of IRGD nanoparticles injected IP. And this translates into improved efficacy when the polymersomes are loaded with paclitaxel, for example. Now, another implication of IRGD-based delivery is co-administration-based delivery. And this takes a little more effort to understand how does it work, but I hope to explain it to you. I mentioned that IRGD peptide binds to neuropilin after being processed by protease. And neuropilin, as you may have heard, is a core receptor of vascular endothelial growth factor. And in fact, it binds to those arginine-ending peptides. The neuropilin binds to arginine-ending peptides. Sorry, I have to go back. And then this binding activates the increase in vascular permeability. So basically, you can think of short peptides like ARPAR peptides shown here as mimics of VEGF, what concerns the effect on vascular permeability. And this is shown here. This is an Evans blue vascular permeability assay. Essentially, what we do is we inject mice with Evans blue dye, which binds with high affinity to circulating albumin. And then we inject into the skin the compounds that we want to evaluate from the point of view of inducing vascular permeability. And as you know, both VEGF and ARPAR peptide induce accumulation of blue color, meaning that vessels become leaky, whereas control compounds don't. Now, in the case of IRGD, the sender peptide is exposed only in tumor tissue. So exact same thing, increase in vascular permeability happens only within the malignant tissue. And this means that the bystander molecules injected in tumor mice along with IRGD peptide start exiting tumor blood vessels more than they would do in the absence of IRGD. And this is illustrated here, again, pancreatic cancer in mice. And here, without IRGD, we see only a small leakiness of tumor vessels, a bit of blue color here. Whereas when we inject Evans blue together with IRGD, we have this massive accumulation of Evans blue, telling that IRGD makes tumor blood vessels leaky. And there is, of course, a lot of mechanistic detail behind it that I have no time to discuss. But essentially, it tells you that now you can use IRGD to potentiate an accumulation and activity of drugs, of systemic drugs. And here is how well it works. So now we have, in this case, breast cancer, autotopic breast cancer in mice, and we inject those mice with Abraxane, which is paclitaxel albumin nanoparticles. And those Abraxane particles are labeled with fluoresceamine, so they are green fluorescent. Untargeted Abraxane shows some tumor accumulation, but relatively weak. Now, IRGD Abraxane conjugate shows very good accumulation in tumors, but also IRGD co-administered with Abraxane accumulates very nicely in tumors. So both conjugated and co-administered delivery modes work nicely with IRGD. And there have been many studies by our and independent labs that have really validated this both co-administration and conjugated delivery mode. In particular, co-administration-based delivery is clinically relevant because it would provide a simple way of potentiating clinically approved drugs. And here is just a list of compounds that have been tested already for IRGD code delivery. And this, as you see, includes compounds such as small molecules, nanoparticles, antibodies, and even cells, and in a variety of tumors. This can be, of course, and is highly clinically relevant. So we decided to found a company that is called Cend Therapeutics. And Cend, of course, is because of this CN rule of neuropilin engagement, and it's pronounced as send. So we are sending something to tumor with our technology. And this company is 5 years old, and it has already been able to take the IRGD into man. Now we are about to enter phase 2 trial in treatment of pancreatic cancer. And what we do is that we co-administer IRGD with gemcitabine and Abraxane. And preliminary data seem quite interesting. Of course, as you know, phase 1 is all about safety and toxicity, but already there, of course, we looked at the response, and it seems that something is happening. Of course, it's too early to make anything further. Now, IRGD, in addition to IRGD tumor-penetrating peptide, now we have identified a number of other peptides that use a similar mechanism. And the theme is that they all have different primary receptors. In case of IRGD, it's integrins. In case of INGR, it's aminopeptidase, then it's p32. But the common theme is that after this initial tumor recruitment, they always proteolytically process to these active arginine or lysine ending sender peptides that are capable of interacting with neuropilin, and then they trigger this vascular exit and tumor penetration pathway. And then, just since it might be relevant to current situation in the world, is that already when we first described those sender peptides, we couldn't help not to notice that many pathogenic viruses display similar sender-like motifs on their surface. And as you know, furin is a protease that is involved in processing of many viral coat proteins, and furin likes to cleave at basic residues at the RXXR motif, which is a sender motif. And after this cleavage, those peptides start binding to neuropilin. And already in our original work, we proposed that those sender mechanism of tissue and cell penetration might be actually involved in spread and tropism of many viruses. Some are brought here in this list. And indeed, just before the SARS pandemic, we started collaboration with colleagues in Munich, Finland, and UK. And and we are well placed to do quick studies on coronavirus 2, SARS coronavirus 2, and our studies show that neuropilin is a receptor and host factor for this virus to infect the cells. And it really could explain some things that have been very hard to explain based on first receptor that we all know about, which is ACE2. And these studies are now under review in a major journal. And I thought to mention it, especially to younger audience here, saying that you kind of, even if you work on cancer, your work might become relevant to something that is far away, but you have to keep a broad view and maybe contribute your reagents and expertise to make an impact. Then I will quickly provide you some examples of our current work. And what we are now working on is on what we call homing peptides version 2.0. So essentially, whereas the first homing peptides that people described were generic tumor homers, usually binding to angiogenic vessels, now we are interested in peptides that would target specific components within the tumor. Would it be, for example, certain tumor-associated immune cells, tumor extracellular matrix, etc. And first example I will show you is PL1, which is a bispecific extracellular matrix targeting peptide. As you know, many tumors are very rich in extracellular matrix, and the matrix is often made not by tumor cells themselves, but by tumor-associated stromal cells. And this means that matrix is, that those cells, of course, are more genetically stable, and the matrix is perhaps a better target than genetically unstable tumor cell displayed molecules. Another aspect is that matrix is highly abundant and allows higher capacity delivery of compounds to tumor. And we focused on these two molecules. One is extra domain B of fibronectin, or EDB, and then the other is trinacin C isoform. And these both modules of extracellular matrix are highly tumor associated and importantly already developed as targets of antibody drug conjugates and immunocytokines by a company in Switzerland and Italy called Philogen. And a PhD student in my lab, Prakash Lingasamy, asked the question, can we target those molecules, these two molecules, EDB and trinacin, with a single peptide? And advantage of being that the single peptide would target both, and being a peptide, it would be a lot cheaper and easier to synthesize and produce and translate than those antibody molecules. So he did a simple biopanning on EDB and trinacin C in cell-free system, then saw a nice enrichment in binding of phage library over the course of biopanning. And then, long story short, he identified a 12-mer peptide that specifically binds to both EDB and trinacin C. And then he moved on and developed nanoparticles for as a model payloads for PL1 targeting. He used iron oxide nanoworms, these elongated iron oxide nanoparticles that are particularly useful for in vivo targeting, and of course are also active as MRI contrast agent, as I showed you a moment ago. And then the second is silver nanoparticles that have also used that I will describe in a moment. So with iron oxide nanoparticles labeled here in green, Prakash saw that PL1 functionalized particles showed very nice homing to tumor blood vessels across a panel of glioma models, and we tested some other tumor types as well, and they were all positive, whereas control non-peptide nanoworms essentially showed no signal. And this tumor homing was specific. We could show that if we pre-injected the mice with blocking antibodies to EDB or tenacin, we reduced the homing, and cocktail of both antibodies essentially abolished the accumulation. Then, you know, maybe some people, some of you have worked with optical imaging, and it's very nice and everything, but in a way, often those optical imaging-based approaches are prone to biases and errors. So next we wanted to develop very reproducible and robust technique to allow us to estimate the homing of the particles. And what we did was that we developed silver nanoparticles made from two isotopes of silver, silver 109 and silver 107. And these are phenotypically identical, except that on one type of particle, we coated the peptide that Prakash identified, and then the other was coated with a control. And then again, we mix these particles one-to-one together, inject them into systemic circulation in tumor mice, and then assess the biodistribution profile of both particles. And what is nice about the system is that you really have this internal built-in control that takes care of intratumoral differences that are so confusing often if you compare one animal with the other. So essentially, now we saw that we get in the tumor tissue much higher amounts of PL1 nanoparticles compared to control particles shown in this laser ablation ICP mass spec image from section here. And then also in this line scan, you appreciate the ratio of silver 109 to 7 that shows that you can have up to 30 times more of targeted particles compared to the control in certain sites in tumor. Whereas in the case of normal tissue, such as liver, the ratio stays similar to input, meaning 1. And then Prakash also showed that you can use PL1 nanoparticles to image tumors using the iron oxide nanoworms. And finally, he used in proof of concept study the nanoworms functionalized with pro-apoptotic peptide with sequence cluck-cluck, with cluck-cluck being the amino acids. And we saw that the PL1 targeted nanoparticles with the apoptotic peptides suppressed tumor growth. Recall how much time I have at the moment. maybe I should start wrapping up.
I don't know, 5 minutes, 10 minutes
Yes.
most, please.
Okay, I do it in 5 minutes, and maybe I show you just quickly another peptide, which is p32 targeting peptide. And this is work of postdoc in my lab, Pille Säälik. And p32 is an interesting protein. It's in normal cells expressed in mitochondria only, and it's not exposed on cell surface, but in activated cells, such as in tumors or atherosclerosis, it gets displayed on the cell surface. And p32 was originally identified as a cell surface marker by past postdoc in Öki's lab, Pirjo Laakkonen. And then one of the last projects that I did as a postdoc was a biopanning on p32 to find this peptide that we call TT1 that binds to p32 better than LIP1. And just to quickly show you some data, the p32 targeting TT1 peptide nanoworms again accumulate very nicely within glioma lesions and other solid tumors as well. And what we saw in this very nice and advanced lentivirally induced GBM model, with which we were helped by Tino Friedman-Morvinsky in Tel Aviv, is tumor accumulation, first tumor macrophage accumulation, which was particularly evident for perivascular macrophages, which are thought to be important for immunosuppression within tumors. And then Tino also saw in her lab that indeed LIN-TT1 nanoworms also accumulate in tumor-derived endothelial cells, which is an important target because, as you might have heard, gliomas are resistant to anti-angiogenic therapies. They become resistant over time. And this is thought to be due to mechanism of tumor cells transdifferentiating into endothelial-like cells. And now we saw that with LIN-TT1, we can target those newly differentiated cells that are both GFP positive, which is from tumor, and CD31 positive. So you can appreciate this structure here. And I think due to combination of this macrophage and tumor endothelial, transdifferentiated endothelial cell targeting, the therapeutic approach using again the pro-apoptotic peptide LIN-TT1 nanoworms was so efficient in prolonging survival of these highly aggressive tumors. And I simply want to leave you with this conclusion or summary that there is a system of vascular zip codes that we use for tumor targeting, but that can be used also for targeting of other diseases or helping in, for example, developing vehicles for crossing blood-brain barrier. That is important part of activity in my lab, and that this diversity can be really agnostically discovered and explored by in vivo phage display. And these peptides can be used to achieve more accumulation of payloads at target sites and less exposure at non-target sites, and increased efficacy. And our hope is that if IRGD clinical development will be successful, then this will be a landmark work that will certainly pave the road for future affinity peptide-based drug candidates. And these are current members of my lab that have contributed to this work, collaborators, and of course, Öki Roslahti, with whom we still collaborate a lot. And I will be happy to take any questions that you have.
Hi, Tambet, thank you very much for your wonderful presentation.
Thank you.
It's a great work. You've been working with different kinds of very difficult tumor for treatments. I mean, glioblastoma, pancreas cancer, and the others you showed, all of them are very difficult for the treatment. It seems that it's a wonderful approach for this kind of cancer treatment. So congratulations, very wonderful work and very good talk. We have a few questions here. Are you listening?
Yes, I am.
Okay, great. We have a question from Dr. Ricardo Krieger. His question is, IRGD seems very promising in mice. Can you tell us a little bit more about the phase 2 results, preliminary results?
Phase 2 is about, I mean, it's just starting now, meaning that we just, we have no data on it. It's starting now in these 4 sites in Australia that I showed you. So basically all we have is phase 1 and some data on, responses from this phase 1. We had there 30, if I'm correct, 34 patients. So already sizable cohort. And of course, there are some interesting things there, but I'm not allowed to disclose it.
All right, but it's one question. It's my question is, when you use this kind of delivery, any kind of delivery, of course, you always has the risk of side effects. It's for different reasons, even if it's too specific. Have you seen this in mice or even in your this phase 1 process, experiments? Or?
So you are saying about side effects?
Using any kind of delivery or treatment. In this case, for this your peptides. So in some cases, it cannot get the target and goes in the liver or goes in the spleen. So even if it's too specific for the tumor or any kind of disease. So have you seen this side effects in your experiments?
I think, especially with co-administration mode, the beautiful part is that the peptide itself is really, completely not toxic. Now we basically take this peptide that, has no side effects on its own, and then combine it with toxic therapy. So it's not like we do a new chemical entity or something. It's simply a peptide itself and Abraxane Gemcitabine by themselves. And then what happens is simply that now you shift the distribution profile of Abraxane and Gemcitabine by peptide. You get, this is again like it varies, but typically you would get anywhere between 2 and 20-fold more payload into tumor with IRGD. And then, of course, your option being a clinician is that you will keep the usual dose of your drug and just combine it with IRGD to more drug in the tumor, or B, you inject less drug into the patient to have side effects and then achieve the same level of drug in tumor that you would get with high dose without IRGD. So you can decide whether to go for lower side effects or higher efficacy, or stay in the middle.
All right. Marcelo, you want to make the next question, please?
Yes, thank you for the presentation, Dr. Martin. I would like to, very nice, I have followed very far the RGD story since 20 years ago. And I'm very happy to see that even a mechanistic stuff, how to penetrate the endothelium, it's very, very interesting. The results is very amazing. I would like to ask whether there is also antibodies are also used to target tissue cells and specific cells, and it can be used to target tumors also. How to compare antibodies to these peptides, these homing peptides, in clinics or as a drug, biotherapeutic? How do you compare antibodies to peptides?
So you are, of course, in clinic, it's very, it's not straightforward to, you could easily do those things, comparative studies in mice, and this is part of the reason we are developing these isotopically tagged particles, for example. Because now, being in this field for 15 years, really one of the big drawbacks is, or disadvantages, as you, over the time has been, as you point out, that it's very hard to put peptides in context. Like every new group reports a peptide, then they say it's fantastic and great and targets everything. So now a big part of our work is kind of auditing those peptides. So we put them, we have now, instead of just 2 silver isotopes, we have more. We also include palladium, taupe, silver, and so on. And then we just have, let's say, a combinations of peptide injected, particles with peptide injected into tumor mice, and then we can get like highly reproducible ratios and homing patterns defined in direct, head-to-head comparison mode. And I think that's a way we should go. It's very hard to do this kind of thing in patients, but at least even in mice, if we can get a good view of value and comparative view, that will help us focusing our efforts.
Okay. And can I have another question? It's very interesting that you showed the mechanism for virus to be explored. Do you think you can use peptides for treating viral infections?
Totally. What we have here is, this is really something exciting, obviously. And there is, you know, what we know, we have identified this small part in neuropilin that binds to sender peptides. The peptide virus is displaying multiple copies of sender, and now with this small neuropilin-derived domain, we can basically wrap virus in to inactivate it, and that's ongoing work. And what makes it particularly beautiful, you know, all these antibodies and so on, they are virus specific. We have accumulating evidence that if we use neuropilin-derived traps of sender, then we can move towards a pan-antiviral. You know, it's almost, maybe it sounds too good to be true, and probably there will be, you know, problems, but basically all this list of viruses I showed you, they can be potentially inhibited by this sender trap. And that could be why it's also so important, is that if you look at genetically unstable viruses like coronavirus, those RNA viruses, then one of the most conserved sites in the genome is the one that encodes for furin cleavage site. This doesn't really change. So it makes like a perfect, virus cannot, you know, move away from it. This, you know, makes it even more interesting. So that's certainly exciting, yes.
Yeah, it's very exciting. Do you are planning some animal tests or something like that, model system?
Yeah, we actually, now we evaluate this system in coronavirus, influenza, like bird flu, semi-liquid forest virus, and some others. And this is, of course, we have no BSL-3 here, but our colleagues have, and they do those tests. We develop those neuropilin-based antiviral candidates.
That's good. Thank you. Thank you very much for your time. And
so there is a last question. Sorry, it's from Dilip Kumar, and he said, "Congratulations for a wonderful presentation. The question is, how can we maximize low affinity distribution of nanoparticles to high affinity in different organs separately in vivo? Is there any particular method?"
You know, like with my training as a biologist, it's mostly like a bit of trial and error. I know there are people like our good friend and collaborator Giuseppe Battaglia, who is a big expert. You know, he's at UCL and he does a lot of biophysical, like modeling, like how can you play with the valency to achieve this or that kind of homing patterns, even with the same peptide. But in our case, the approach really is that we do it diagnostically. We have a phage particle, it's 50 nanometers, it displays about 200 peptides. And then we translate this into the like clinically relevant or experimental particles with same density. So phage, if phage works, then our thinking is also this particle has to work. And then guys like Giuseppe then take it forward and say what happens if we, you know, modulate the number of peptides, what happens with homing pattern. And this is like entirely a huge field and important field, I agree.
Okay, thank you very much, Dambe. Marcelo, you have any other question? Okay, thank you very much for this kind and wonderful talk. Congratulations for your work. I hope you can come to Brazil on May next year. So yeah, so.
Absolutely. Let's hope things get back to what they were. Yeah.
All right, hope so.
Thank you very much.
Thank you very much.
Thank you.
And see you soon.
Bye-bye.
Hope so. Bye.